Dear students, welcome to Time class. So this is your biology session. So welcome to the biology session. I believe this is your very first biology session, and in this session we will discuss the basics 1, that is chapter 1. What is the name of our chapter? That is cell, tissues and body movements. So let me ask you one thing. Do you like biology? How the subject biology for you? So why it is, I think it is probably easy for you to learn the concepts of biology, isn't it? Why is it so? Why is it so? Because biology means what? What do you mean by biology? The term biology can be divided into two, that is bios and logos. It is derived from two words. Bios means life, logos means knowledge. So we can say biology is the study of living organisms. It is all about life. It is all about ourselves. It's all about the organisms, the plants, animals, different microorganisms, whatever living things are surrounding us in this planet Earth, that is known as biology. Okay, it is a very lively subject. It is also very interesting subject, and we still it is the science is fascinating us by unveiling the truth of life on Earth. So moving on to the session, I would start with the first topic, that is, what is our first topic? Cell, the basic unit of life. Cell, so we need to know what a cell is. Why we are calling the cell as a basic unit of life. We all are living organisms, isn't it? We all are living organisms, and we have some peculiarities. Like, what are the peculiarities of a living organisms? So we can grow, we can reproduce, we can multiply, and we all have the capacity to respond to stimuli. We all have the capacity to perform all the basic life processes in our life. We all can move, and we all are capable of locomotion, isn't it? Yes. So we will be studying about some features of what a cell is, who discovered cell, how a cell looks like, and do all types of cells looks alike, or what are the constitution of a cell, what are the things inside a cell, that we will be discussing. Okay. So how are you, students? I hope you all are doing good and you all are keen to listen to me. So listen here. We are going to talking about the discovery of a cell as an interaction. You need to know what is the history behind discovery of a cell. So what is the story behind the discovery of a cell? So a scientist named Robert Hooke in 1665. Who was Robert Hooke? He was a English botanist. Botanist means what? The person who study about plants. Okay, so the cell was first discovered by Robert Hooke in 1665. How he came up with that particular word, that is cell? Because when he was examining a thin slice of cork under a primitive microscope and observed a structure similar to honeycomb of many little compartments, so he called this as a cell. What is the meaning of the cell? The cell meaning in Latin, it is a little room. So what is the thing? What's the story about this cell? That is, Robert Hooke was a scientist. He was born in England, London. So while he was observing a thin slice of cork means what? It's a thin section of the bark of a tree under a primitive microscope. At that time, our new-gen compound microscope or electron microscope or such a sophisticated microscopes were not invented. Okay, so he self-defined a microscope and he observed the structure like what? Which is similar to a honeycomb. Have you seen a honeycomb? Yes, how it looks like? It looks like little compartments. So he called the little compartments as a cell. Why he give the name as a cell? Because the cell meaning little room in Latin, isn't it? So cellular is the little room of the in monasteries. There will be a little room which was inhabited by monks. Okay, so he gave the name as the cell. Okay, this is the history of a cell. Then in 1674, another scientist known as Anton van Leeuwenhoek, please note the name Anton van Leeuwenhoek, observed free living cells in pond water because at the time when Robert Hooke discovered a cell, he couldn't see anything living inside the cell. And it is Anton van Leeuwenhoek who discovered the free living cells in pond water in an algae known as Spirogyra. Okay, yes. So cell was discovered by Robert Hooke. The credit goes to him. But the free living cells, because Robert Hooke couldn't describe about whether there is a cytoplasm or whether there is a nucleus, or he didn't know whether any living components were present inside that little compartments. So later on, Anton van Leeuwenhoek first discovered the free living cells in pond water, in an algae. So, cell is the basic structural and functional unit of life. For example, when we compare our body to a building, we can compare our body to a building. So, for making a building, for construction of a building, we need what? The blocks or bricks, little units. Like little units, like blocks or bricks, together constitute a complete building or an infrastructure. Is that clear? Yes, like that. Every organisms are made of the structural. So, they are made of cell, and that cell itself is the functional unit of life. Then, all living organisms are made of cells, whether an organism which have only a single cell as their body. Okay. Then, sometimes a single cell may constitute an organism, and in other case, an organism consists of many cells. Sometimes, in the case of amoeba, the single cell itself is their body. In our case, whether we have millions and millions of cells. Okay, so we have multicellular organisms. We have many more cells. So, organisms can be of a single cell, or in other case it consists of many cells. Is that clear? Yes, it is all about introductory part. Then, according to this, organisms can be divided into two types. According to what? According to the number of the cells, organisms can be divided into two types. What are the types? They are unicellular organisms and organisms with only one cell. What are unicellular organisms? You can, when you look into that word, unicellular. Uni means what? Single-celled organisms. They are unicellular organisms. The organisms with only one cell. Example, what are the examples for unicellular organisms? They are amoeba, chlamydomonas, they are paramecium, and bacteria. These all are the examples of unicellular organism. Then about multicellular organisms, organisms with many cells. Multi means what? Many. So multicellular means many-celled organisms. Organisms with many cells, example plants and animals. Then what do you mean by cell theory? What is cell theory? The cell theory was proposed by Matthias Jacob Schleiden. Okay, we can simply call him as Schleiden. Okay, the cell theory was proposed by who? Or Matthias Jacob Schleiden, 1838, and Theodor Schwann, 1839, and was later modified by Rudolf Virchow. Cell theory was put forward by Schleiden and Schwann, and it was remodified. It was modified by another scientist, that is Rudolf Virchow. So that name of the scientist you have to keep in your mind. Okay. Then the main postulates are, what are the main findings, or what are the main concepts of a cell theory? What are they? All organisms are made of cells. All organisms are made of cells. That is the first postulate or the first point coming under cell theory. Then moving on to second point, cell is the basic structural and functional unit of life. That means an organisms are made of cell and they are functioning on the basis of the functions of a cell. Then all cells arise from pre-existing cells, only. That means all new cells arise from the what cells? That is old cells. That is the meaning of that sentence. All cells arise from pre-existing cells only. Then what is this? Omnicellular e-cellular. Omnicellular e-cellular means, it is the Latin form of that sentence. Omnicellular e-cellular. All cells arise from pre-existing cells. So first 2 points, what is the first point? All living organisms are made of cells. Cell is the basic structural and functional unit of life. And these 2 points are put forward by, or these 2 points are contributed by Schleiden and Schwann. Later the third point, that is all cells arising from pre-existing cells, was put forward by whom? That is Rudolf Virchow. So that is the point, that it is modified by Rudolf Virchow. Then scientists and their contributions. So what are this? We have to keep in your mind the name of the scientist. Name of the scientist is Robert Hooke, that is discovered cell. So when a question comes, who discovered cell? You can answer it as Robert Hooke. So once again, I will recap the introductory part. It was Robert Hooke who discovered cell. Okay. And later, Anton van Leeuwenhoek, another scientist who first discovered or first observed the living cell in pond water. Then, who is Robert Brown? So we have to note down the difference between Robert Hooke and Robert Brown. Robert Brown discovered the nucleus inside a cell. That is it written as discovered cell nucleus. Then what is Purkinje? Who is Purkinje? Purkinje is the name of a scientist. He's a scientist. He coined the term protoplasm. What is that? He coined the term protoplasm. And what is protoplasm and what is nucleus, you will be learning in the following sessions. So coined the term. So who coined the term protoplasm? Purkinje. Then who proposed cell theory? Cell theory was proposed by Schleiden, Schwann and Virchow. So shortly it is given as Schleiden, Schwann and it is modified by Rudolf Virchow. Then Camillo Golgi. Camillo Golgi is the name of a scientist and he was the first person who first described the Golgi apparatus, who was the first person to describe about Golgi apparatus. Golgi apparatus is a cell organelle which is found inside the cell. And what is its structure and what is its function, that we will discuss it to the later part of the session. So, Moving on to next point, the size of a cell. So before going to size of a cell, what all things we have studied? What all things we have discussed very now? Who discovered cell? It was Robert Hooke in 1665. In a cork, when he observed the cork of a tree, that is bark of a tree, through a primitive microscope of a self-defined microscope, he was able to see little compartments, and he called it as a cell. What is the meaning of a cell? Meaning of a cell is a little room in Latin. Later, it is Anton van Leeuwenhoek who discovered the living cell in pond water in an algae known as Spirogyra. And depending upon the number of the cells, organisms can be divided into unicellular and multicellular. Okay. Then again, about cell theory. Cell theory was proposed by whom? It was proposed by Schleiden and Schwann. And later it was modified by Rudolf Virchow. That are things we have discussed very now. Then size of cell. What about size of cell? Most cells are minute and are visible only under microscope, isn't it? Yes, it is very minute, so it cannot be visualized through our naked eye. It should be visible only under a microscope. Then they are only few micrometers in diameters. That means what? They are only they are minute structures, few micrometers. Micrometers means it is a very small part of a meter in diameter. Then the smallest cell is mycoplasma. Which is the smallest cell? The smallest cell is mycoplasma, that is 0.3 m, whereas the largest one is the ostrich egg. Okay. Then the longest cell is the neuron or nerve cell. So smallest cell is mycoplasma, and whereas the largest one is the ostrich egg. Which is the longest cell? Longest cell is the neuron. Then coming to shape of cell. So depending upon the function of a cell, the shape of the cell may vary. So there are different types of cell based upon their shapes. So the shape of the cell varies according to their function. There may be what all shapes they can possess. They may be disc-like or polygonal in shape or columnar in shape or cuboidal in shape, or they may be thread-like structures. That all things. So in the previous slide, we have discussed about what size of the cell, which is the longest cell, which is the smallest cell, and which is the largest cell, and what is the shape of the cell. Then nerve cell has an elongated structure. So nerve cell is the longest cell, isn't it? So nerve cell has an elongated structure. It is lengthy, and this shape helps them to conduct the impulse quickly. So since it is lengthy, so since the nerve cell is elongated in structure, so this structure helps them to perform their function. So what is the function? It helps them to conduct or transmit the impulses quickly. That is, what is meant by impulses? That is, when we respond to a stimuli. So it is transmitted from one cell to another cell in the form of impulses, in the form of ions, positive and negative ions. So when we respond to a stimuli, it is traveling in the form of impulses. That is a function of a nerve cell. Then similarly, there are cells whose shape is flexible, like amoeba and some blood cells. So there are other cells whose structure is not a rigid, it is flexible in shape, and so they can move into, so they can get into different shapes they want. So it is shapeless. And the flexibility in shape helps amoeba to perform various activities like. So since the shape of the amoeba is flexible, do you know what amoeba is? Yes, everyone can draw amoeba, isn't it? A person who is not even know how to draw anything, everyone can draw amoeba. Because why? Because it doesn't have a typical shape of its own. It will form into different shapes and size according to the need of the body. So flexibility in shape helps amoeba to perform various activities like movement, feeding, etc. The change in shape of amoeba is due to the formation of pseudopodia, which are projections on the body. So how the amoeba is changing its shape? Because its cytoplasm inside the body, when it moves to a particular direction, we can see some foot-like structures. Okay, foot-like structures, but they are not original foot like ours. It is pseudopodia. Pseudo means what? What's the meaning of the word pseudo? Pseudo means it is false. Podia means feet or foot. Okay, so it is a false foot. So they are the projections of the body of amoeba. Truly, they are not having an all different legs or limbs like ours. So that is the pseudopodia can be seen in amoeba. That's all about the shape of the size of the cell. Then what is the shape of a cell? So what is the shape of a cell? The shape of a cell may vary. You can see that is in case of red blood cell, it is round and it is a biconcave in shape. That is round in size. What is biconcave in shape? What is the shape of a concave? There are two types of lenses, convex lens and concave lens. Concave lens, its edges will be what? Its edges will be bulged or it is round. Concave means what? Its edges should be forming inward. Okay, edges should be not bulged. It is depressed inside. So that is meant by round and biconcave. Two sides it is concave in shape. Then what about white blood cells? We have two types of blood cells, that is RBC and WBC. RBC is known as erythrocytes and WBC is known as leukocytes. Okay, so white blood cells, that is what is the amoeboid in shape, that is it doesn't have a perfect shape. Look at these structures. Yes. Then columnar epithelial cells, their shape is long and narrow. Columnar epithelial cells which are found in the internal organs of our body. So what is its shape? It is long and narrow in shape. Okay. Then columnar epithelial cells can be seen in the internal organs and digestive system. In the intestine of our stomach is lined by columnar epithelial cells. Then what about this nerve cell? Nerve cell, it is branched and long. How it looks like? It is not a single one, it is branched. It has a cell, a nucleus, dendrons, dendrites, and it is axon like that. When you study the structure of a neuron, you will be studying what are the different parts of a neuron. So here the figure is conveying the message to you that there are different shape for a cell depending upon their function. It is their function which decides their shape. Okay. Then what are the different types of cell? Type of cell. The cell can be prokaryotic cell and eukaryotic cell. There are two types of cells, prokaryotic cell and eukaryotic cell, according to the difference in cellular organization. So what is the difference between prokaryotic cell and eukaryotic cell? That is, prokaryotic cells are primitive and lack a true nucleus. So it is very important, that is very basic facts to know about a cell. What is prokaryotic cell? Prokaryotic cell means the word pro means primitive. That is karyotic means nucleus. That means it have a primitive nucleus. Prokaryotic cells are primitive and they lack a true nucleus. Why it is telling that? true nucleus. So for knowing what is a true nucleus, you need to study about the structure of a cell. So that is the difference between a prokaryotic cell and a eukaryotic cell. Eukaryotic cell, they will be having a true nucleus, and prokaryotic cells, they lack a true nucleus. Then another difference is their genetic material is not surrounded by nuclear membrane, and it is confined to a region called nucleus. That is, in all the cells, we should have a genetic material. What is genetic material? In some organism, it will be DNA. In some organism, it will be RNA. In some viruses and all, genetic material is RNA. And in most of the bacteria, the genetic material is DNA, deoxyribonucleic acid. And this nucleus, in the case of ours, that is we human beings are what we are eukaryotic organism. So our genetic material is kept or located inside the what? Nucleus, and this nucleus will have a well surrounded a double walled nuclear membrane is present. But in the case of prokaryotic cell, they are not having a true nucleus. That is, their nucleus doesn't have a nuclear membrane surrounded by it. They will be having a genetic material. So that particular region without a nuclear membrane can be called as nucleoid. Okay, then what is a prokaryotic cell structure? That is, this is structure of a typical structure of a bacteria. You can say bacteria is a typical example of a prokaryotic cell. What are its parts? We can see a flagellum for movement, and first of all, this is the nucleoid region. That is a region where the chromosome, the genetic material, whether it be DNA in the case of bacteria. So this DNA is not surrounded by a nuclear membrane. It is freely suspended in the cytoplasm. So that region is known as nucleoid. So prokaryotic cell is having a nucleoid region. Then beyond that, they are having a plasma membrane, and outer to that, there is a cell wall. Okay, and they have a cell organelle, that is ribosome, is present. Okay, so these all are the parts of a prokaryotic cell. That is typical structure of a bacteria is shown here. The outer wall is known as cell wall. Inner to that cell wall, this yellow colored cell wall, inner to that plasma membrane is present, and in the middle the genetic material, that is DNA, is just suspended in the cytoplasm. It is nucleoid, and ribosomes are they are the cell organelles, and flagellum is the movement for locomotion, for movement, and this is the cytoplasm. So these all are the parts of a prokaryotic cell. A typical bacteria structure is shown here. Then cells are generally smaller than eukaryotic cells. So eukaryotic cells itself will be smaller than that prokaryotic cells will be. Then there are four basic shapes for prokaryotes. For bacteria, what are they? That is, it may be of spherical shape, then it is known as coccus bacteria or cocci. Example, Staphylococcus. Then bacillus, it is a rod-shaped bacterium. Example, lactobacillus bacteria, which is seen in curd. Then Vibrio, if it is a comma shaped, it is known as a Vibrio bacteria. Example, Vibrio cholerae, that is a bacteria causing disease cholera. Then if it is a spiral shaped, we call it as spirillum. Okay, spirillum. Example, Trypanosoma. So depending upon the shapes, we call it as different names. If it is spherical shaped, it can be called as a coccus bacteria. So the category of bacteria which is having spherical shape are included under the group that is coccus. If it is rod shaped, if it is known as the group of bacteria, bacillus, bacilli. Typical example is lactobacillus. Then if it is a comma shaped, we call it as a Vibrio, for example, Vibrio cholerae. And if it is a spiral shaped, we call it as a spirillum, and example, Treponema. So these are the shapes. This is the coccus, and this bacillus, it is Vibrio, and this is spirilla, spiral shaped. All cells possess the common structure. That is, depending upon its functions, cells, the shape and size may vary, but it has a common structure. The common structures which is found in different cells, like cytoplasm is present in all cells, cell membrane is present, cell wall is present, like that, features are common. Then all prokaryotic cells have, what are they? They have a cell wall. All the prokaryotic cells possess a cell wall. What is a cell wall? A cell wall is the outer layer, and it provides integrity to the cell. That is, in the case of plant cells, the cell wall is present. Okay, in prokaryotic cells also cell wall is present. So it gives the provide an integrity. It forms as a boundary of the cell from its outer environment. So it also gives mechanical strength to the cells because they are made of peptidoglycans. Peptidoglycans are in turn made of carbohydrates. They are carbohydrate units. And it is absent in mycoplasmas. That is, mycoplasma is a group of organisms coming under prokaryotes, and they are the smallest non-living cell, and many of the mycoplasmas are pathogens in plants and animals. What is the peculiarity? Most of the mycoplasmas are they do not have a cell wall, and they can survive without oxygen. So they are pathogenic also. Pathogenic means what? They are disease causing. Then moving on to next structure, that is plasma membrane. That is, first structure is outer layer, that is cell wall. Second one is plasma membrane. It lies just inside the cell wall. So when I show you the structure of the bacterium, I showed you that a structure which is inner to the cell wall, that is plasma membrane. They are made of lipids and proteins. So plasma membranes are made of lipids and proteins. Cell wall is made up of peptidoglycans. So you have to note all these things. Okay. Then what about cytoplasm? Then cytoplasm is the fluid substance present inside the cell. Inside the cell, a fluid substance is there. It is known as cytoplasm. And what is its peculiarity? It lacks all kinds of membrane-bound cell organelles. So in the cytoplasm of a cell organelle, it doesn't have the membrane-bound cell organelles. Cell organelles are of two types. A membrane bound, that is, most of the cell organelles are bounded by a membrane. Okay, so they are known as membrane-bound cell organelles. But in prokaryotic organisms, the membrane-bound cell organelles are absent. Membrane-bound cell organelles, like we will study, that is lysosomes, Golgi bodies, mitochondria, plastids, vacuoles, like that. But you have noticed a structure, that is ribosomes is present. That is, ribosome is a non-membrane-bound organelle. Okay. So only ribosomes are present as an organelle in prokaryote. Then what is mesosome? Mesosomes are formed by plasma membrane extensions into the cell. So mesosomes they are not a separate cell organelle or unit. They are the part which is the extensions of the inward invaginations. The inner invaginations of what? Cell membrane or plasma membrane in the cell of the prokaryotic. Then what is their function? They help in processes such as cell wall formation and respiration. Okay, so here you can see this is the cell wall. Inner to it, plasma membrane is present, and the plasma membrane have some form of extensions, the invaginations or inward extensions into the cytoplasm, and they are known as mesosome or mesosome sac. What is its function? Because they help in the process of cell wall formation, and it also increases the surface area of the cell membrane and where the respiration take place. What is respiration? Respiration means the breaking down of food substances to produce energy. Okay, that is respiration. So it is they are the site of respiration also. So that's about mesosome. So cell wall is present, plasma membrane is present, cytoplasm, and mesosomes are present in prokaryotic cell. Then about ribosomes. So you notice that is a non-membrane bound structure, cell organelle, that is ribosomes are present inside the prokaryotic cell. The only cell organelle found to be present. Then they are the sites of protein synthesis. That means what? Inside the ribosomes, the protein synthesis, the production of proteins take place. Then these ribosomes, they are found associated with plasma membrane. So what is that? They are always been found associated to the plasma membrane, not bound by any membrane system. So it is not separately bound by any other membrane system. They are found associated with plasma membrane. Who is associated with? Ribosomes are associated with plasma membrane. Then next thing, that is cytoplasmic inclusions. What is cytoplasmic inclusions? There are some bodies, inclusion bodies, lie free in the cytoplasm. So it is not compulsory it should be present in all prokaryotic organisms. It should be present only in particular prokaryotic cells. Then what is the function of this cytoplasmic inclusions? They help in the storage of reserve materials. Some materials which are found to be in excess should be stored in the cytoplasmic inclusions for the future use of the cell. So, for example, phosphate granules are such a thing to be stored inside the cytoplasmic inclusions. So, that's all about ribosomes and cytoplasmic inclusions. Then, moving on to eukaryotic cells. So, we have discussed about prokaryotic cell, what is their peculiarity and all. And here we will going to talk about that is eukaryotic cell. So what is eukaryotic cell? Eu means what? True. Karyon means nucleus. So they have a what nucleus? They have a true nucleus, a well-defined nuclear membrane. Inside the nuclear membrane of the nucleus, the genetic material DNA is present. What are the eukaryotic cells? These eukaryotic cells include, or eukaryotic organisms consist of protista, plants, fungi, and animals. What are they? Protista, plants, fungi, and animals. These are the classification of these organisms, that is protista, plants, fungi, and animals. Kingdom, the living organisms are categorized under 5 kingdoms. Kingdom Monera, Kingdom Protista, Kingdom Fungi, and Plantae, and Animalia. So Monera, it consists of bacteria section, that is eubacteria and archaebacteria. So bacteria will come under prokaryotic cell. All other kingdoms like Protista, Plantae, Fungi, and Animalia are included under eukaryotes. Okay. Then eukaryotes have a true nucleus, that is their genetic material is surrounded by a nuclear envelope. They have a true nucleus, that means it's surrounded by a nuclear envelope. They possess membrane-bound organelles like endoplasmic reticulum. What are the membrane-bound organelles? Endoplasmic reticulum, mitochondria, Golgi borders, etc. So, non-membrane bound or cell organelle, an example is one and only ribosome. Membrane bound organelles are endoplasmic reticulum, mitochondria, Golgi borders. Then, animal cells and plant cells show considerable variations in their structure because there are typical differences between an animal cell and plant cell. So, what is the very important and what notable difference between the plant cell and animal cell is that plant cells are surrounded by an outer cell wall, whereas cell wall is absent in animal cell. That is a very important point to be noted. That is, plant cells have a cell wall. Cell wall is present in plant cells, whereas in animal cell, the cell wall is absent. Then similarly, plastids and large, that is, plastids means the plastids are cell organelles which contain the colored pigments like chlorophyll. Then they have different other pigments inside it like oil, starch, carbohydrates, like that. So plastids and a large central vacuole are present in plants and not in animals. Okay, so what is the difference between plant cell and animal cell? Animal cells do not have a cell wall, but plant cells do have a cell wall. Then plastids is absent in animal cell and also a large central vacuole, which is a storage organelle for the plants, is absent in the case of animal cells. Between prokaryotic cell and a eukaryotic cell. So the size generally small in the case of prokaryotic 1 to 10 micrometer and size generally large when compared to prokaryotic cell, that is 5 to 100 micrometer. And a true nucleus is absent in the case of prokaryotic cell, and in eukaryotic cell a true nucleus is present. And in prokaryotic cell, it contains a single chromosome and it is more than one chromosome in a eukaryotic cell. And membrane-bound cell organelles like endoplasmic reticulum, Golgi complex, etc. are absent in prokaryotic cell. In membrane-bound cell organelles are present in case of eukaryotic cell. So this is the differences and it is very important. And moving on to cell wall, plant cells are surrounded by a non-living, rigid, permeable outer layer called a cell wall. So the plant cells have a cell wall, isn't it? When compared to animal cell, they do not have. So plant cells are surrounded by, it's a non-living thing and it is a rigid and it is also permeable also. Permeable means it allows the substances to move in and out of the cell. So what is function? It provides structural integrity, and that is rigidity and protections to the cell. It gives a mechanical strength. They are made of cellulose. It is very important. Cell wall is made up of cellulose. And cell walls of adjacent cells are joined together by a jelly-like substances called middle lamella. So the adjacent cells, they are joined together by means of middle lamellae. That is the features about cell wall. They are made of cellulose. It is the most important point. And cell membrane, they are made of proteins and lipids. So the cell membrane is made up of proteins and lipids. And semi-permeable, that is, they are selectively permeable or semi-permeable. What do you mean by semi or selectively permeable? They do allow the exit and entry of molecules, but not all the molecules, only selected molecules. So it is known as semi-permeable membrane, allowing entry and exits of only some materials. So this is the structure of a membrane, that is cell membrane. It is a microscopic view of a cell membrane. It has a lipid bilayer, that is the lipid, that is outer and inner layer, so that it is known as bilayer. It has two layers of lipid, so it is named as lipid bilayer. And in between the lipid layer you can see some type of proteins, that is integral proteins, membrane bound proteins are present. And this is another shape of the protein. The shape of the proteins may vary. Okay, so this is the structure of the cell membrane. They are made of proteins, and proteins are of different kinds, and they also have a lipid bilayer. Then moving on to next about protoplasm. What is meant by protoplasm? It is the living material consisting of cytoplasm. So inside the cell, cytoplasm is present, and cytoplasm together with nucleus and all other cell organelles is called protoplasm. So protoplasm is the living material. So for a cell, outer covering is, for a plant cell wall, inner to it cell membrane is present, inner to it a fluid like a substance, that is cytoplasm, is present. Cytoplasm together with nucleus and other cell organelles, all together we call it as a protoplasm. Okay, so we can tell like this, that is protoplasm is equal to cytoplasm, nucleus and other organelles. What is cytoplasm? Cytoplasm is the fluid content, the only fluid content. Okay, the fluid content inside the cell membrane. It contains many membrane bound organelles. Then what is cell organelles? Moving on to cell organelles. An organelle is a organelle meaning a little organ. Okay, is a unit inside the cell which perform a special function. So there are different kinds of cell organelles. Is that inside the cell there is the division of labor. A cell inside the cell, there are different organelles. So the division of labor is happening inside the cell. Each cell organelle are assigned a particular work for them. So they are doing their jobs independently. Then the various functions performed by the cell are actually divided among the organelles. There is a division of labor inside the cell. For example, energy production is the duty of mitochondria. That is, it is performed by mitochondria, and photosynthesis is performed by plastids. Okay, and how it is performing the photosynthesis, that we will discuss when we discuss about plastids. Then the various cytoplasmic organelles are given below. So we are going to discuss in detail each and every cell organelles. What are they? They are endoplasmic reticulum. What are they? They are, first one is endoplasmic reticulum. Please note, we are going to start discussing about endoplasmic reticulum. We have now discussed all about the outer structures, that is cell wall, cell membrane, inner to it protoplasm is present, and the fluid content is cytoplasm, which contains many cell organelles, and each cell organelle is having different jobs. Different jobs are assigned to them. That means there is a division of labor inside the cell. So first one is endoplasmic reticulum. What is endoplasmic reticulum? It is a large network of reticulum of membrane bound tiny tubular structures. So it is a membranous structures. So it is in the shape of a tubular structure. That is the peculiarity of endoplasmic reticulum. It is a membrane system. It's a part of the membrane system. Then they are found scattered in the cytoplasm. So majority of the cytoplasm will be occupied by means of what? Different cell organelles, and one of the cell organelles is endoplasmic reticulum. Then endoplasmic reticulum is of two types. How they are differentiating into two types? That is, there are two types, that is rough endoplasmic reticulum, that is abbreviated as RER, rough endoplasmic reticulum, and another one is smooth endoplasmic reticulum. Why it is calling by its name rough endoplasmic reticulum? Because of its appearance. because on this rough endoplasmic reticulum, on the endoplasmic reticulum which is having a ribosomes, they are known as rough endoplasmic reticulum. They possess ribosomes on their surface. They are helping in protein synthesis. What is smooth endoplasmic reticulum? It is abbreviated as SER. They lack ribosomes, so their surface will be smooth. So it is not a smooth endoplasmic reticulum. It lacks ribosomes on their surface. So rough endoplasmic reticulum have a rough look appearance on its outer surface because of the presence of ribosomes, which helps in protein synthesis. And smooth endoplasmic reticulum, it lacks ribosomes on their surface, and they are the main site for lipid synthesis. What is lipid? It is fatty acid and glycerol. That is lipid. Fats and oils are the examples of lipids, are coming under lipids. They are also involved in the detoxification of poison. That is, poisons or drugs which are harmful to our body or toxins. That detoxification is going inside the smooth endoplasmic reticulum. So we have to note the difference between rough and smooth endoplasmic reticulum. Ribosomes. So what is ribosomes, or what is ribosomes looks like? They are dense. They may be dense. It is spherical particles. They are present in both prokaryotic and eukaryotic cells. So we have to note the point that which is the cell organelle which is present in both prokaryotic and eukaryotic cell. That is ribosomes. Why they are present? Because they are not membrane bound. They are attached to the plasma membrane. So it can be found in both prokaryotic and eukaryotic organisms. They are the sites of protein manufactures. The proteins are manufactured or synthesized inside the ribosomes, and they are found either in cytoplasm or they are found attached to the rough endoplasmic reticulum. That's why it gets its name RER, isn't it? Then not bound, they are not bound by any membrane, and they are made of complex of RNA molecules. It is a single-stranded genetic material, and they are known as rRNA. That is ribosomal ribonucleic acid, ribosomal RNA. And proteins, they are also made of protein subunits, that is ribosomal proteins. So ribosomes are made of complex of RNA, and it is a protein. They are made up of RNA molecules and also protein molecules. That is ribosomal proteins and ribosomal RNA, rRNA and R proteins. Protein subunits, larger subunits and smaller subunits. There are two subunits for the proteins. Then moving on to Golgi apparatus. What is that? Golgi apparatus. So endoplasmic reticulum is over and ribosomes are over and Golgi apparatus. Now we are going to talk about Golgi apparatus. It was first described by Camillo Golgi in 1898. So what is the shape? It consists of a system of many flat disc-like structure. I will show you the structure. That is like this. So it consists of many flat disc-like disc-shaped sacs called cisternae. Okay, what is this? Cisternae, which are arranged parallel to each other. And what is its function? That is the proteins which are synthesized in the ribosomes which are attached to the rough endoplasmic reticulum. It is package such type of substances, whatever substances which are produced inside the endoplasmic reticulum or inside the nucleus, and it is packaged and it is stored and dispatched by various products which is received from endoplasmic reticulum. So what is its function of Golgi apparatus? That is, so the cell can be compared to that of a mini factory, isn't it? In some cell organelles, that is, substances are produced And if you are manufacturing any substances in a factory, like a clothing or footwears or food materials, whatever it may be, it should be properly packaged. Packaged means some addition of substances should be done and some removal of substances, or it should be properly packed. For example, the proteins which are formed, which are produced inside the ribosomes, will be in a long strand. It should be properly packaged into its primary, secondary, tertiary, or quaternary structures by means of which apparatus? Golgi apparatus or Golgi bodies we can call. So that is the function. Packaging, storing and dispatch of various products are done by Golgi apparatus, which are received from the endoplasmic reticulum, and it remains in close association with endoplasmic reticulum. Golgi apparatus, because these substances have to be packaged and stored, so it need to be lie near to the endoplasmic reticulum. It involved in the synthesis of complex sugars and lysosomes. What is its function? They are involved in the synthesis of complex sugars and lysosomes. So note the point. Lysosome is another cell organelles which have been synthesized inside Golgi apparatus, and some other complex sugars are also produced inside the Golgi apparatus. We have come to an end of this session, that is our topic cell, the basic unit of life and its cell organelles. So cell organelles and its following parts should be continuing in the next sessions, and now for the time limit we will stop here. Before stopping, we just mash up or just revise the topics we have just discussed. Okay, of life, and after that we have discussed, that is introduction, the cell, its meaning, little room. It was discovered by Robert Hooke. Then it looks like a honeycomb or many little compartments, and it is Anton van Leeuwenhoek who observed free living cells in pond water in 1674, and cell is the basic structural and functional unit of life, and all organisms are made of cells, and sometimes a single cell may constitute an organism. In other case, many cells are also present. Then, depending upon the number of cells, organisms can be divided into unicellular and multicellular, and cell theory was proposed by Schleiden and Schwann, and it is later modified by Rudolf Virchow. So what are the main postulates? All organisms are made of cells. And next one is cell is the basic and structural functional unit of life. All cells arise from the pre-existing cells, and here we discussed the different scientists, that is Robert Hooke discovered cell, Anton van Leeuwenhoek observed first living cell, Robert Bow discovered cell nucleus, Purkinje the term protoplasm was coined, and cell theory was proposed by Schleiden, Schwann and Virchow, and it is Camillo Golgi who first described Golgi apparatus. Then we have discussed about the size of the cell. Most cells are minute, they are microscopic in structure. The smallest cell is the mycoplasma, and the largest cell is the ostrich egg. Longest cell is the neuron, and the shape of the cells varies according to the function, and they may be of different shapes like polygonal, columnar, cuboid or thread-like. Okay, then nerve cells has an elongated structure. Nerve cells have an elongated structure, and it may be in the shape of a flexible shape like that of amoeba or blood cells, and amoeba have certain false foot-like structures known as pseudopodia, which are the mere projections of their body's cytoplasm. Then in the next slide we have seen different cells, red RBC, WBC, columnar epithelial cells and nerve cells. So they have different functions. To perform that function, they have different shapes. Okay. Then what are different types of cell? They are prokaryotic cell and eukaryotic cell, and they have true nucleus in eukaryotic cell. Prokaryotic cell doesn't have a true nucleus, they have nucleoid. Then we have discussed the structure of a cell, that is a prokaryotic cell having a nucleoid, plasma membrane, cell wall, and ribosomes, flagellum, and the cytoplasm. Then we have seen the prokaryotic, that is the for basic cell, coccus, bacillus, vibrio, and spirillum. So they have different names based on their different structure. They have different categories. These are the different shapes. Then, though the shape they varies, prokaryotes have a cell wall, they have outer layer, it is the integrity and rigidity to the cell, and cell wall is made up of peptidoglycan in the case of bacteria. Okay. And mycoplasma, that is cell wall, is absent. In plasma membrane, it lies just inside the cell wall. They are made of lipids and proteins, and cytoplasm is a fluid-like substance. And mesosome, they are the mere extensions of plasma membrane, which helps in cell wall formation and respiration. Then what we have seen, we have talked about ribosomes. They are the site of protein synthesis, and they are not bound by any membrane system. Okay. Then cytoplasmic inclusions, they lie free in the cytoplasm. Their function is storage. Then eukaryotic cell examples, kingdoms like protista, plants, fungi, and animals, they have true nucleus, hence the name eukaryotics. And cell organelles like ER, endoplasmic reticulum, mitochondria, and Golgi bodies are present. Animal cells and plant cells show considerable variations in the structure. So, what are variations? Plant cells do have a cell wall, animal cells do not have. Plastids and central vacuoles are present in the case of plant cell, and in animal cell it is absent. And this is the differences between prokaryotic and eukaryotic. Okay. Then cell wall, plant cells, they are non-living, rigid, permeable. They are made of cellulose. Their strength is mechanical, giving mechanical strength and rigidity to the cell. And what about cell membrane? They are made of proteins and lipids. And they are semi-permeable cell, also known as selectively permeable, because it allows only the entry and exits of only some materials. This is the structure. And we have seen protoplasm and cytoplasm. It's a fluid content. Protoplasm means all the things inside the cell. That is cytoplasm, nucleus, and other organelles. Cytoplasm is the fluid alone part. And we are seeing about cell organelles. There is a division of labor because different cell organelles perform different function. Okay. Then about endoplasmic reticulum, it's a large network of membrane system which is scattered inside the cytoplasm. It's of two types, RER and SER. In RER, ribosomes are present. They are rough. In smooth RR, there is no. In smooth endoplasmic reticulum, there is no ribosomes, and function is lipid synthesis and detoxification of poisons. And we have seen talk about ribosome. What is peculiarity? They are the sites of protein synthesis, and in both prokaryotic and eukaryotic cell, ribosomes are present, and they are made of complex RNA molecules and proteins. And Golgi apparatus, first discovered by Camillo Golgi. It consists of what? Cisternae, that is flat disc-like structure which lie parallel to each other, and these functions include the packaging, storing, and dispatch of various products. And it always remain associated with endoplasmic reticulum. They are involved in the synthesis of complex sugars and lysosomes. So, with this Golgi apparatus, we are stopping this session, and in the next session we have an exercise section. We are discussing the question. We should able to know how much you have understood. So we will be seeing in the next session. Thank you, students. I hope you have enjoyed this session. Thank you and have a wonderful day.